Hi everyone, my name is Martin. I'm an application specialist at BMG LabTech. Today I'm here to talk to you about possible approaches to determine binding affinity on microplate readers and which advantages our microplate readers offer for this purpose. The content of my talk will be a brief showcase of binding interactions and why they are relevant, which detection techniques are commonly used for the investigation, and how to perform binding studies and determine binding affinity. Finally, I will show you two examples from our application note library which highlight binding studies. Binding interactions are crucial events which rely on the proximity of two biomolecules to each other. Without interactions, there would be no enzymatic activity, no receptor signaling, no cellular transport, and no DNA transcription or translation. Life would not be possible without them. But how do they work? Binding interactions are made possible by various different forces which allow intermolecular connections such as van der Waals forces, hydrogen bonding, electrostatic forces, and hydrophobic effects. While most interactions are non-covalent and reversible, some binding interactions are based on covalent bonds that are formed between biomolecules. So why are binding interactions relevant? If they are misregulated, they are often cause of disease. This can range from misregulated enzymatic activity, misregulated signal transduction, development of autoimmunity, to misregulated gene expression, leading to disorders such as diabetes or Alzheimer's. Thus, binding interactions are an important aspect of drug research. Drugs are often specifically designed to target specific binding interactions. Furthermore, drug discovery studies large libraries of potential small molecules for potential therapeutics. These have to be as potent and as specific as possible to prevent undesirable side effects. Kinetic parameters such as binding affinity are crucial tools for this purpose. Common targets for drugs comprise kinases and GPCRs, which are the focus of many studies, but also other protein-protein interactions regarding the proteasome or other proteases. So now that we know why they are relevant, how can we actually monitor binding events? Here I would like to highlight 4 different approaches commonly used in binding studies. First, FRET and TIA-FRET, which are both based on the energy transfer between two fluorophores. These fluorophores are attached to the two binding partners of a binding interaction. Since they have overlapping emission and excitation spectra, emission of the donor fluorophore leads to the subsequent excitation of the acceptor fluorophore, when both are in close proximity. Time-resolved FRET uses long-lived lanthanate donor fluorophores to reduce background signals even further. BRET methods work in a similar way. However, in this approach, the donor signal is provided by a luciferase, which generates emission light by an enzymatic reaction. As with FRET, BRET assays are performed as ratiometric measurements of acceptor and donor signal, offering comparability between different test runs. Fluorescence polarization assays use polarized excitation light and also measure the polarization of emission light to investigate a biomolecule. This method uses small fluorescent ligands which on their own have a rapid tumbling rate. Accordingly, the polarized excitation light becomes depolarized. After binding to a larger biomolecule, the tumbling rate of the fluorescent ligand is reduced and its emission light retains most of its polarization. Comparing the polarization of unbound to bound fluorescent ligand allows to perform binding studies. The final approach I would like to present today is AlphaScreen. This method uses donor and acceptor beads which are bound to two interaction partners of interest. When in proximity, energy transfer from one bead to the other leads to the generation of a luminescent light signal, which can then be quantified. What is special about this method is that the excitation light has a longer wavelength than the measured emission signal. Now that we know about different approaches to measure binding interactions, how do we actually perform binding studies and determine binding affinity? The first step is to understand that binding is not static. The interaction of two biomolecules is comprised of association events according to their association rate and dissociation events according to their dissociation rate. Binding changes over time until it reaches an equilibrium where the concentrations of binding complexes is constant. Accordingly, binding studies are performed over time as so-called binding kinetics. Here, three parameters are particularly important. K-on, the association rate or on rate, defines how much of a ligand L binds to a biomolecule B over a specific time to form biomolecule ligand complex BL. K-off, the dissociation or off rate, on the other hand, defines the amount of ligand that dissociates from the biomolecule ligand complex over a specific time. Together they form the equilibrium dissociation constant Kd of a ligand, which is the ratio of dissociation over association events over time. Kd indicates the affinity of a ligand to a biomolecule and is an important parameter for binding and drug research. The lower the Kd value, the higher the affinity of a ligand or drug to its target, which means lower ligand concentrations are needed to induce binding. However, since high affinity can be the cause of either a slow K-off rate or a high K-on rate, it is important to determine these parameters to fully understand a drug's or ligand's binding behavior and choose the right administration. Dependent on the investigated biomolecules, desired readout, and kinetic parameters, different approaches can be followed to perform binding studies. In the following section, I would like to highlight three different approaches to determine the binding affinity of a ligand. First, and most straightforward, is the determination of binding affinity by performing saturation experiments. Here, a ligand of interest is titrated across a range of concentrations to a fixed amount of its target binding partner. By plotting the generated signal of the binding complex over time against the increasing ligand concentrations, Kd can be graphically determined as the concentration of ligand at which half the ligand binding sites of a biomolecule are occupied at equilibrium. While this approach is straightforward, it is important to note that it can be restricted by the assay capacity and is not applicable in all cases. Dissociation and association experiments are the more commonly used techniques to determine the binding affinity of a labeled ligand. Here, Kd is calculated from the determined K-off and K-on rates of a ligand and its target biomolecule. Here the first step is to determine the dissociation rate K-off of the labeled ligand. For this purpose, labeled ligand, here depicted as red dots, is incubated with labeled receptor until binding equilibrium is reached. The measured signal is hereby dependent on the formation of binding complex. To start the dissociation experiment, vast amounts of non-labeled ligand, here depicted as blue dots, are added, which results in the displacement of labeled ligand from the target biomolecule. This results in a continuous decrease of binding signal over time and allows the calculation of the K-off rate of the labeled ligand in per second. After the determination of K-off, association experiments can be performed to determine the association rate constant K-on. For this purpose, increasing concentrations of labeled ligand, again depicted as red dots, are mixed with labeled biomolecules, depicted as purple squares. The time course of association is then observed by measuring the binding signal increase over time in the presence of rising labeled ligand concentrations. This results in a number of association binding curves, as can be seen in the graph on the right side. The corresponding data can be fitted together with the previously determined K-off value to calculate the association rate constant K-on of the ligand in per molar per second. Association and dissociation experiments can even be performed right after each other in one measurement approach to determine both K-on and K-off. For this, first an association experiment is performed until labeled ligand and labeled target have reached equilibrium. Then an excess of non-labeled ligand is added, leading to continuous displacement of labeled ligand. K-on and K-off are determined by calculation. In contrast to sole association experiments, this approach provides reliable fit values for both K-on and K-off, even through only one ligand concentration is employed. But what to do if your ligand of interest is not labeled at all? Can you determine its binding parameters regardless? The answer is yes, by performing competitive binding experiments. However, for this you need a previously established assay system with the labeled target biomolecule depicted as purple squares and an established labeled ligand with known kinetic parameters K-on and K-off. To perform the experiment, increasing concentrations of the non-labeled competitor are added to a fixed concentration of target biomolecule, simultaneous with a fixed concentration of labeled ligand. Here, the addition of increasing competitor concentrations in measurement samples leads to the subsequent decrease of binding signal. By keeping the labeled ligand parameters fixed, the fitting procedure provides K-on and K-off rates for the compound of interest. An alternative way to calculate KD for a non-labeled competitor is by the IC50 method. For this, only the KD value of labeled ligand and the labeled target biomolecule are needed. Again, addition of increasing concentrations of non-labeled competitor lead to a decrease in binding signal. By fitting these data to an appropriate fit model, the IC50 can be calculated, representing the amount of competitor which displaces 50% of labeled ligand from the target biomolecule. With the IC50 of the competitor and the used concentration of labeled ligand and its KD, the KD of the competitor can be calculated with the Chang-Prosoff equation. While this approach allows the accurate calculation of KD for the competitor, it does not provide K-on and K-off values, which should be taken into account for planning your binding studies. Now that we know what different approaches may be used to determine the binding affinity of a ligand of interest to a target of interest, I would like to show you some examples from the BMG application note library, which you can access for free via our website at bmglabtech.com. Application note 348 is a nice example for the previously presented methods to determine binding affinity. It highlights a TR-FRET-based binding assay for the investigation of kinase binding. This homogeneous assay format was ideally suited to screen for kinase inhibitors in high throughput. In a first step, the kinetic parameters K-on and K-off of a labeled tracer were characterized with a combined association and dissociation measurement. Next, 270 non-labeled kinase inhibitors were screened for kinase binding using the previously determined parameters of the fluorescent tracer ligand. Potent inhibitors such as staurosporine led to a concentration-dependent decrease of binding signal over time. For this approach, our Ferrostar FSX was used, which comes with special features for the study of binding affinities. A special feature for TR-FRET approaches is the availability of a dedicated laser for TRF. Compared to xenon lamps, such lasers yield high excitation energy at a specific wavelength, leading to improved assay performance and lower limits of detection. And such lasers are also available for alpha technology. Our Ferrostar reader bases its measurement on so-called optic modules, which contain all the filters you need for your measurement, including excitation and emission filters and dichroic mirrors dedicated for your specific assay. The Ferrostar can be equipped with 4 dedicated photomultiplier tubes on top of the dichroic mirror setup, which allows to split the signal light beam in half and measure 2 emission wavelengths at once, exactly at the same time, rather than measuring them sequentially, one after the other, with the same detector. This cuts measurement time for such assays in half and also increases data quality, since the 2 signals derive from the same excitation event and are less susceptible to other potential influencing variations. Since some binding events happen very quickly, it's best to add some components right before or even during the measurement. This way you won't miss possible binding events and can observe their complete time course. In the present application note, the Ferrostar utilized our integrated reagent injectors. While labeled tracer and unlabeled competitors were already present in the wells, terbium labeled kinase was added with the reagent injectors, and samples were read directly after their addition. Installing up to 2 reagent injectors per BMG reader allow you to flexibly select injection speed and volume while your reagents are kept light protected in the reagent reservoirs. The second example for performing binding studies in microplate format I would like to highlight today is our application note 346, which deals with the study of PROTACs. These bifunctional molecules are designed to bind to a target protein of interest and an E3 ubiquitin ligase. The resulting complex of PROTAC, target protein, and E3 ligase leads to the ubiquitination and subsequent degradation of the target protein by the proteasome. Accordingly, PROTACs have a high potential as therapeutics to regulate transcription factors or proteins implicated in disease. Here, PROTAC activity was monitored by using cells expressing ligbit and BRD4 high bit. After the expression, these molecules with high affinity to each other spontaneously form the nanoluciferase attached to BRD4. To monitor the formation of ternary ubiquitination complex with the target biomolecule, cells were additionally transfected to express halotag VHL, while ubiquitination was monitored by cells expressing halotag ubiquitin. This allowed this kinetic study of both ternary complex formation and ubiquitination in the presence of increasing PROTAC concentrations and screening for PROTAC efficacy. As you can see, ubiquitination starts a little bit after the initiation of ternary complex formation as expected. I would also like to mention here that this assay took place in living cells. To keep your cells happy during the whole measurement time of your experiments, BMG LabTech offers incubation and shaking options. Our readers offer temperature incubation from room temperature up to 65 degrees Celsius, depending on the equipment of the reader. Furthermore, the internal carbon dioxide and oxygen concentrations inside the reader can be monitored and regulated with our atmospheric control unit. With BMG LabTech readers, you can also choose between different shaking patterns, which can be adjusted from 100 to 800 rounds per minute. Last but not least, our mass data analysis software offers very nice features for the evaluation of binding events, next to other features like the wizard for standard curves. Automated template functions allow you to analyze test runs according to your predefined wishes with one click, making the evaluation of big data sets extremely easy and convenient. Kinetic curves and advanced binding kinetic fit models are also available. The software is FDA 21 Part 11 compliant and offers easy Excel, ASCII and graphic export. With this, I would like to come to the end of today's presentation. Thank you very much for your attention. For more information on microplate-based applications, feel free to contact me and the team at applications@bmglabtech.com or visit our website for our extensive library of application examples.